In this video, we'll talk about fertilization in sea urchins. So, sea urchins are sea-dwelling creatures, so they're swimming around, sperm is swimming around in salt water. When sperm gets near an egg, the first thing that they're going to get close to is the jelly coat of that egg. Within the jelly coat is the vitelline layer. Within the vitelline layer is the plasma membrane. Coming out of both the plasma membrane and vitelline layer are a bunch of bindin receptors. So what we're going to look at in this video, though, is how the sperm, which has egg recognition proteins sticking out of its surface, interacts with this jelly coat, changes its shape so that bindins, which are located actually inside the sperm to start, end up on the surface of the sperm, interact with this vitelline layer. What the jelly coat and egg recognition protein and bindin receptor interactions provide is the prevention of cross-species fertilization. All right, so I'm going to bring up now a picture. First, here's a photograph of a bunch of sea urchin sperm stained with I forget what. Here's my comic of a sea urchin sperm anatomy. This is the sea urchin's sperm's plasma membrane. This is the acrosomal membrane here. Here is part of the jelly coat of the egg, what is it, a fusican sulfate protein or something like that. I forget exactly what it is, but it's a sugar sulfate that the egg recognition proteins recognize. So, when that interaction occurs, either directly or now thought to be pretty indirectly, sodium channels are opened. And when sodium channels are opened, that of course is going to change the membrane potential of the cell. What's it going to do? It's going to depolarize the membrane. Depolarization of the membrane, what's that going to do? Well, it's clear, since that's labeled voltage gated, that it's going to open the calcium channels. But something that surprises me is there seems to be a non-calcium mediated and more direct membrane potential change in the activity of adenylate cyclase. And adenylate cyclase, as you remember, is the enzyme that takes ATP and turns it into cyclic AMP. Cyclic AMP then turns around and activates this sodium proton antiporter. Why is this important? Okay, sodium coming in, allowing calcium to come in. Calcium coming in leads to the exocytosis of the acrosome with calcium signaling. And the protons leaving raises the pH inside this cell. That pH rising allows actin polymerization to occur. So now let's take a more macroscopic look at what we're depicting here. Here is the resultant exocytosis of this acrosome. So that's the acrosome exocytosing. And this is, I think it's called lysine or something. It's a Y. lysine is going to eat through the jelly coat. I think it's also going to help eat through the vitelline layer when it gets there. Then these bindins, which were on the inside surface of the acrosome, which was a big vesicle that exocytosed, are now, you'll see, since this membrane is continuous here, you see that this set of bindins is now on the surface of this plasma membrane of the sperm. And because the pH has gone up, the actin monomers that were in the actin body become actin polymers. Big long rods are formed, and that pushes the front of the sperm forward, and this is called the acrosomal process. So it's kind of strange. That's the acrosomal process, both because that's what the set of events is called, but it's also what that long, stringy looking thing right there. There's your acrosomal process right there, and here's the sperm proper. So now notice that those bindins are growing, they're being pushed towards the very tip of the egg. When they get to the tip of the egg, that leads to a bindin receptor interaction. So I guess we got to go way back to that first diagram. Bindin receptor. I'm going to get rid of this other one. And bindin meeting with the bindin receptor here's the process, here's the bindin sticking out, and here's the receptor. This is a very species-specific relationship. So the bindin and receptor is a very successful block to wrong species or cross-species fertilization. The jelly coat interaction was also a block to wrong species, cross-species fertilization, but a less vigorous one, a less exacting blockage. Okay, so in this video, all we've looked at is the acrosomal reaction, which allows the head of the sperm to get to the vitelline layer. We haven't had fertilization yet, but we have shown the two levels of cross-species fertilization prevention in this process, and shown how the sperm can get close. And in the next video, we'll talk about how it gets the rest of the way to the actual egg membrane where fusion occurs and the actual fertilization event.